Number 1, selective breeding produces more offspring, fewer offspring, or desired traits in offspring. Number 2, the crossing of buffalo and cattle to produce beefalo is an example of inbreeding, hybridization, or transformation. Number 3, between which nucleotides is the DNA cut in Figure 13.1? A, between adenine and uracil. C, between thymine and cytosine. Or D, between guanine and adenine. So take a look at what broke off on the DNA up there, and that'll tell you where it's cut. Number 4, one function of gel electrophoresis is to A, separate DNA fragments for comparison, B, copy large quantities of DNA, or C, recombine DNA. Number 5, a DNA molecule produced by combining DNA from different sources is known as a hybrid, a polyploid, or recombinant DNA. Number 6, in an electrophoresis gel sample, band B is closer to the positive end of the gel than band A. Which of the following statements is true? A, band B is more negatively charged than band A. B, band B moved faster than band A. Or C, band A is smaller than band B. Number 7, during transformation, A, a prokaryote is changed into a eukaryote. B, a cell takes in DNA from outside the cell. Or D, a cell is mutated. Number 8, sorry for the movement. Number 8, which of the following can be observed in a karyotype? B, an extra chromosome. C, genes. Or D, alleles. Which can you see in a karyotype? Number 9, in a pedigree, a circle represents B, a female, C, a child, or D, an adult. Number 10, a pedigree can be used to A, determine whether a trait is inherited, B, show how a trait is passed from one generation to the next, C, determine whether an allele is dominant or recessive, or D, all of the above. Number 11, which of the following genotypes result in the same phenotype? Number 12, most sex-linked genes are located on A, the autosomes, B, the X chromosome only, or C, the Y chromosome only. Number 13, the formation of a Barr body, A, causes the genes on one of the X chromosomes in a female cell to be switched off. B, always causes the same X chromosome in a female cells to be switched off. Or C, switches on the Y chromosome in a male cell. Number 14, if non-disjunction occurs during meiosis, A, only 2 gametes may form instead of 4. B, some gametes may have an extra copy of some genes, or D, it occurs during prophase. Number 15. Scientists test for alleles that cause human genetic disorders by A, making karyotypes, C, looking for the DNA sequences that occur in the alleles for those disorders, or D, by making pedigrees. Number 16. The process of DNA fingerprinting is based on the fact that B, no two people, except identical twins, have exactly the same DNA, C, most genes are dominant, or D, most people have DNA that contains repeats. Number 17. The Human Genome Project is an attempt to A, make a DNA fingerprint of every person's DNA, B, sequence all human DNA, or D, identify alleles in human DNA that are recessive. Number 18. The purpose of gene therapy is to A, cure genetic disorders, B, determine the sequences of genes, or D, change dominant alleles into recessive alleles. Number 19. Gene therapy is successful if the A, viruses carrying the replacement gene infect the person's cells, C, if the replacement gene synthesizes a desired protein in the person's cells, or D, if the replacement gene is successfully spliced to viral DNA. Number 20. Which of the following fields of genetic engineering requires actual manipulation of DNA? B, inbreeding, C, cloning, or D, mutation? Number 21. Which type of genetic engineering involves the use of a single adult cell to produce a genetically identical individual? A, hybridization, B, cloning, or C, transformation? Number 22. To produce transgenic bacteria or transgenic bacteria that make insulin, which of the following steps did scientists have to do first? A, insert the human insulin gene into a plasmid, C, use a restriction enzyme to cut out the insulin gene from human DNA, or D, transform bacteria with the recombinant plasmid.